Can you see those slides?
Yeah.
Okay, cool. think we'll just wait 1 more minute, see if anyone else joins, and then we'll get started. Okay, I think we'll get started. So I hope you're expecting a lecture on embryonic development. There's quite a lot of ground to cover, so I'm going to do my best to hit your spec points as best as possible. Let me know if I'm going too fast, and you can email me with that email on the screen if you have any questions at any point. Okay, so I thought when I was learning it for the first time that embryology was a bit difficult. And so I thought before we got into the details, we'd look through some key terms first because they're going to be used throughout. So it's good to have a bit of background. So it can be quite difficult to orientate yourself in the 3D space that's talked about in embryology. And so this picture here shows how the different axes are labeled. So there's two main ones, rostral caudal, and that's from your head to your toes, with rostral being your head and caudal being your toes, and dorsal ventral. So that's like front to back, and ventral is the front and dorsal is the back. Another key concept is potency, and this relates to how many cell types a cell is able to become. So a totipotent cell can become any cell type, including the placenta. And as development goes on, the fate of cells typically becomes more restricted, all the way down to unipotency or terminally differentiated cells that can only be like one cell type. Next up are some key terms linked to patterning. So this is the way cell fate is determined. Induction is the term for the way cells dictate the fate of other cells, and they do that by releasing signaling molecules. So these signaling molecules diffuse to cells, and the cells have receptors for them, and then on binding, a signaling cascade occurs that changes patterns of gene expression to alter cell fate. So signaling molecules are conserved in development, so the same ones are used over and over again, but for different patterning events. A morphogen is a special form of signaling molecule, so it acts in a concentration-dependent manner. So that means the pattern of gene expression it causes changes depending on their concentration. And finally, transcription factors act to up and down regulate gene expression, and these are often the very end of these signaling cascades. So fertilization. This is where development begins, and it occurs in the ampullary region of the oviduct. And it's the process by which the haploid nucleus of the egg and sperm fuse to make a diploid nucleus, and this is a fertilized egg, also called a zygote, and it's totipotent at this stage. Now, the process of fertilization can go wrong sometimes, and one of the ways is through the formation of this type of mole. And this is linked to something called genomic imprinting and pronuclear non-equivalence, which basically means the genes essential for embryonic development are silenced in the paternal genome, and the genes essential for placental development are silenced in the maternal genome. So if an embryo is derived from only paternal genomes due to loss of the female pronucleus, then only placental tissue will develop. And this leads to high levels of HCG, and this can produce either benign or malignant tumors in the mother. Okay, so prior to implantation, there are several developmental stages that occur. So these are occurring as there's passage along the oviduct and into the uterus. The first one is cleavage. And in this, the zygote undergoes cell division, but there is no cell growth. And it is thought this could relate to a lack of nutrition being available at this point. But given the lack of cell growth, this is not a sustainable event, so it does not happen for a long period of time. And as this process of cleavage is happening, there's loss of totipotency, so cell fate is becoming more restricted. The next stage of development is blastocyst formation. So that's the picture on the right. And there's an inner cell mass, and this inner cell mass basically becomes the embryo, and then the surrounding trophoblast is vital for placental development. And in between these two, there's a fluid-filled cavity called the blastocele. Blastocysts can be important in terms of twins. So if you have non-identical twins, and they'll be derived from two different zygotes, you will always have two blastocysts. But if you have monozygotic twins, so from one zygote, they can have one or two blastocysts, and that can impact placental development. So the next step is implantation, and this occurs roughly 5 to 6 days post-fertilization. And as we said, the trophoblast is the really important part for this. It embeds and invades into the uterine lining, and this triggers something called the decidual reaction, in which the endometrium becomes more edematous and vascular. And from this point onwards, the process of placental development occurs, which is quite complicated and involves the formation of villi, but due to time constraints, we're not going to go further into that, but you can email me if you have any questions. So it's really important that this process of implantation occurs in the correct place. So you want it to occur in the uterus. Ectopic implantation is when it occurs somewhere else, and this can be quite dangerous. So it most commonly occurs in the fallopian tubes, and this can rupture, leading to hemorrhage in the mother, which is a surgical emergency. So whilst this trophoblast is focusing on implantation and placental development, the inner cell mass of the blastocyst is also changing, and it forms the bilaminar germ disc, which is made of two things, epiblast and hypoblast. So the epiblast is pluripotent, and most of the cells of the body develop from it. And it's continuous with the amniotic cavity, so you can see it in that picture below in blue. And the hypoblast is continuous with the primitive yolk sac, and this was previously the blastocyst cavity. So one of the things I found challenging was understanding the various cavities that form during embryonic development. So far we've talked about the one on the left, the blastocele, and also this one at the bottom here, which has the amniotic cavity at the front and the primitive yolk sac, which was previously termed the blastocyst cavity. So after this, the chorionic cavity begins to form. So this occurs initially through expansion of some mesoderm behind the primitive yolk sac. And then a space appears called the chorionic cavity. The chorionic cavity then expands and acts to pinch the yolk sac, such that there's the formation of the definitive yolk sac from the primitive yolk sac and a connecting stalk, and the connecting stalk becomes the umbilical cord. I hope that made sense. Right, so next up is gastrulation, and this is one of the very important processes in embryonic development. It's one I have seen on previous exam papers, so I thought we'd spend a bit of time here. So this is the process by which the bilaminar germ disc becomes 3 layers, the primary germ layers. But it's also when patterning of the cells occurs to generate the body plan. So in terms of what actually happens, the primitive streak forms, and this is from a buildup of epiblast cells, and so does the primitive node. And then the epiblast of the bilaminar germ disc undergoes epithelial to mesenchymal transition, which allows the cells to become more mobile. These mobile cells then migrate through the primitive streak. The first cells that migrate mix with the hypoblast to generate the primary germ layer called the endoderm. The next cells to undergo this migration become the mesoderm, and finally the epiblast cells that don't undergo the migration form the ectoderm. Germ cells, so those that become the sperm and the egg, are a separate line of cells, but you don't really need to know much about those for your exams. So as this migration process is occurring, the primitive node is releasing signals that instruct and determine the fate of cells, and we're going to talk about that in a couple of slides. But first up, we're going to talk about the primary germ layers. So as we said, the process of gastrulation gives rise to these primary germ layers, and what these layers themselves give rise to often comes up on MCQs, so it's quite a good idea to learn. So the endoderm was the layer that was formed first, and this becomes the lining of the gut tube and associated organs, including, importantly, the lungs, which can often be forgotten about. The ectoderm is the last one to form, and this creates the skin and the neural tissues, and the neural tissues of both the central and peripheral nervous system. Neural crest cells are important derivatives of ectoderm, And we're going to talk about them more in some detail later, but it's just important to remember that they're derived from the ectoderm. The mesoderm layer is probably the most complicated. It gives rise to lots of structures, and it is divided into several things. So in blue, in the picture on the right, you have the axial mesoderm, and that forms the notochord. And then in the reddish color, you have the paraxial mesoderm that forms the somites, and these contribute to the segmental body plan. And then in green is this intermediate mesoderm, which is important for gonad and kidney development. And finally, you have this lateral plate mesoderm in purple, and this can be split into splanchnic and somatic. So somatic is important in limb bud and body wall formation, and splanchnic in visceral wall formation and in the gut and the heart. So I said we'd come back to this, and the second key aspect of gastrulation is the patterning of cells to make the body plan, and the primitive node is central to this. So the experiment on the left is mentioned quite a lot. Essentially, Spearman and Mangold transplanted some of the primitive node equivalent, called the organizer in this species, to the other side of the embryo. So there was an embryo that had two organizers, and this led to duplication of the body axis, and it showed that the organizer, which is the node in humans, was telling the surrounding cells what to do. So this essentially highlighted the role of the primitive node. Now, the patterning of the body plan occurs in all the axes in slightly different ways. So dorsal ventral axis is patterned by signals released from the node in induction. The rostral caudal axis is patterned based on the time through which a cell passes through the node. So the earlier a cell migrates through the node, the more rostral a structure it goes on to develop, and this is linked to Hox gene expression. Left to right patterning is a bit more complex, and it's based on cilia in the node beating to make a flow of liquid that is sensed by other cilia, and this causes different gene expression on the left and right side of the embryo. Now, this process of patterning can go wrong in several ways. So firstly, you can have conjoined twins, and this occurs when the node splits in humans in a similar way to how Spearman and Mangold did it. You can also have a condition called situs inversus, and this occurs when the left to right patterning is incorrect, and it can be either complete or incomplete. So incomplete, everything is switched over, so what's normally on the right is on the left and vice versa. But in incomplete, it's a bit of a mixed match, and there can be more complications. There can also be failure of the streak and node to regress at the end of gastrulation, and this results in something called a sacrococcygeal teratoma, which is a tumor containing lots of different cell types because the node is capable of instructing lots of different cell types. Okay, so next up is the process of embryonic folding. And this is the way that the flat trilaminar germ disc that we've got at the moment with the 3 layers becomes a 3D cylindrical structure that more resembles a human being. So when I was learning this for the first time, I found it quite hard to visualize what was going on. And one of the ways it clicked for me, and you could try it yourselves later, is I got a piece of paper and I folded it in the same way that the embryo does. And it helped me visualize what was going on in a 3D space. I thought it was quite useful. So the folding occurs in 2 main ways. You have lateral folding, and you also have rostral caudal folding. So lateral folding, which is on the left here, results in the formation of the gut tube and the amniotic fluid now surrounding the embryo. So the gut tube is this bit here in yellow, and it's surrounded by the splanchnic lateral plate mesoderm, and the body wall, which is, I don't know if you can see my mouse, but that bit there is surrounded by somatic mesoderm. Now this tube is open at the rostral end, where it will form the mouth, and is also open at the caudal end in something called the cloacal membrane, and this is the anus and urogenital opening. So defects in this lateral folding can lead to failure of the body wall to form correctly, such that the abdominal contents that should be lying inside ends up lying on the outside. And this leads to conditions called a memphysele and gastroschisis, and this, which one it is, depends on whether the organs are membrane covered or not. So they are covered in a memphocele, but they are not in gastroschisis. Rostral caudal folding is the other sort of folding. So rostral folding brings the more rostral structures, like the cardiogenic region, which develops into the heart, the septum transversum, which develops into the diaphragm, and the primitive periocardial cavity ventrally, so to the front. And caudal folding moves the coacal membrane, which is the anus and urogenital opening, and the allantoid that becomes the bladder ventrally, so to the front as well. So all of this folding and the expansion of the amniotic cavity also leads to the formation of the umbilical cord as well, which can be seen in the bottom diagram on the right, sorry. So the next step of development is the separation of the different cavities in the human. So before this, it's basically one cylindrical tube, and this needs to be divided into the abdomen and thorax, and then the thorax split even further into the pleural and pericardial cavities. So separation of the thorax from the abdomen is mostly done by the diaphragm, and most of the diaphragm is made from mesoderm, called the septum transversum, and this originally develops more rostrally than the heart, and rostral folding brings it underneath the heart region. So here it can start to separate the abdominal cavity from the thorax. The separation is complete by several different structures, which I don't think you need to know, but they are the pleural peritoneal membranes, the body wall musculature, and the esophageal mesentery. Now, if the diaphragm fails to form correctly, such that the abdomen and thorax are not separated, you can get a congenital diaphragmatic hernia, in which there's gut contents where the lung would normally lie, and that can be seen on that image of the baby there. So now we've got an abdomen and a thorax, we need to split the thorax into the pleural and pericardial regions. So these are separated by the pleural pericardial folds that develop on either side, and then they fuse in the midline to separate the cavities. Okay, so that was an overview of core processes, and now we're going to have a look at some specific organ systems and their development. So first up is the nervous system. So there are some elements of nervous system development in your spec, and it's particularly focused on the peripheral nervous system. So you'll cover the central nervous system next year. It's more of a second year topic. But there are several things we need to think about in terms of the peripheral nervous system, and the steps are labeled on the slide. So first up is neural induction, and we sort of already covered this. So this occurs during the process of gastrulation, and as we mentioned, the ectoderm that form can become nervous system tissue or skin. And neural induction refers to setting the fate of some of the ectoderm to the nervous system, and they're called neuroectoderm. And it forms a neural plate. And because this is done in gastrulation, it's done by the primitive node. So the next step of nervous system development is something called neurulation. And this is the process by which the neural plate, so the ectodermal cells that will become the nervous system, create the neural tube. So the notochord is really important to this. So the notochord was that structure formed from the axial mesoderm, and it induces neurulation. So firstly, the neural plate buckles at the midline to form something called the neural groove. So the nervous system tissue nearest the notochord is ventral, and this ventral region forms the motor regions of the spinal cord. And this occurs due to patterning from the notochord by sonic hedgehog, or SHH. Next up, we have convergence on the other side. So the dorsal sides of the neural plate come together, forming a tube, and this dorsal part of the tube forms the sensory part of the spinal cord. At this point of fusion, some neuroectoderm is pinched off and becomes the neural crest cells, and there's more to come on those a bit later, but it's just important to know where they come from. So once we've created this neural tube, we have an open cylindrical tube, and the top and bottom need to be closed off. The top and bottom are called neuropores, and the rostral one, so that closest to the head, closes at roughly day 28 of development, and the caudal one a bit later on day 27. Neuropore closure is really important, and if it goes wrong, then there can be a range of different outcomes. So what happens depends on whether it is the top neuropore or the bottom neuropore that fails to close. If the top neuropore fails to close, you get a condition called anencephaly, in which the baby lacks part of the brain and skull, And sadly, babies with this condition will often pass away within a few hours to days after birth. If the bottom neuropore fails to close, then you can get a condition called spina bifida. There are a number of different forms of spina bifida called occulta and cystica, and this cystica can be subdivided into meningoceal and meningomyelocele. It's not just neuropore closure that's important to spina bifida, but also vertebral development. And that's something we're going to talk about later, but it's just important to mention now. So spina bifida occulta occurs in the lower lumbar of sacral region, and it's linked to incomplete vertebral arch development. There are often no symptoms apart from a tuft of hair. Cystica, however, which is more linked to these neural tube closure issues, can have more serious consequences. So in meningocele, the coverings of the spinal cord, so the meninges, pass through the opening in the spine to form a cyst-like swelling in the back. In meningomyocele, it's not only the meninges, but also the spinal cord itself that lies within this cyst, and that's the most serious type of spina bifida. It can affect the pelvic organs, the legs, and also cerebral spinal fluid flow. Right, so we mentioned the neural crest cells a few times, so now we're going to talk about them in a bit more detail. So these were generated from the dorsal aspect of the neural tube, and they undergo epithelial to mesenchymal transition again, so they're able to migrate and they're more mobile. Now, these cells become lots of things in the body, and that is shown on this slide, but there are a few key ones of note. So firstly, they become the cells of the peripheral nervous system. They also form melanocytes, and they also form the facial skeleton. So failure of these cells to migrate correctly and develop can be seen in different ways, depending on the cell type that is affected. So firstly, you have something called Hirschsprung's disease. And this occurs when the enteric or gut neurons derived from the neural crest cells don't develop. The lack of cells in the colon means that stool can get stuck, leading to bowel obstruction. Secondly, melanocytes, which are the melanin-producing cells responsible for pigmentation, can fail to migrate properly too. And this will be seen in issues in terms of pigmentation. So one example of which is the piebald trait, which can be seen on the slide. So the next step of peripheral nervous system development relates to somites. Now, as we mentioned, the somites are derived from the coaxial mesoderm, and they form in pairs from day 20 of development in a rostral to caudal direction. You ultimately get 4 occipital, 8 cervical, 5 lumbar, 5 sacral, and 8 to 12 coccygeal. And they make quite a few structures. So they can be split into the sclerotome and the dermamyotome. The dermamyotome is all linked to the name, so this forms the dermis, connective tissue and fat, and the musculature of the limbs and body walls. And the sclerotome forms the vertebra. So that's what we're going to focus on now. So the sclerotome cells from the somite migrate to surround the notochord and the neural tube. The nerves of the peripheral nervous system are from the neural crest cells, as we said, and they develop in a segmental way. Each sclerotome splits in two around a spinal nerve. So the upper half of one sclerotome and the lower half of another combine, and they go on to form the vertebral body, vertebral arch, and annulus fibrosus. And the notochord goes on to form the nucleus pulposus, so that's also part of the intervertebral disc. Now, this process of vertebral body development can go wrong, and if the sclerotome fails to migrate on one side, then you can get a condition called lateral sclerosis. And this occurs because you get a hemivertebrae in which one side of the vertebrae is missing, and so the spine isn't straight. It leans to one side or the other. Okay, so that's it for nervous system development. Next up, we have limb development. So limb development occurs between weeks 4 and 8 of development. And by the 8th week, you have the basic limb structure all developed. And after this, the elements that are formed just need to grow and mature in size. Now, the lower limb is 1 to 2 days behind the upper limb in terms of development, but the key events are actually very similar. So the first step in limb development is formation of the limb bud. So the limb bud is made of mesenchyme from the somatic lateral plate mesoderm, and it has an ectodermal covering that becomes skin. The next step is hyaline cartilage model development. So you get a cartilage model of the bones laid down within the limb bud, and that's finished by week 6. This cartilaginous model undergoes endochondral ossification to become bone. By birth, the center of bones is normally completely ossified, but at the ends of bone, the epiphyses, which are key to growth postnatally, there is still a lot of cartilage, and ossification doesn't finish there until well after birth. Next up is muscle development. So myoblasts from the somite mesoderm invade and form the muscle mass of the limbs. And then the final important gross anatomical change is rotation of the limbs around the long axis. So this occurs between weeks 6 and 8 of development, and it's quite different between the upper and lower limb. So the upper limb undergoes a modest lateral rotation, whereas the lower limb undergoes a really big lateral rotation. It essentially rotates 180 degrees. And that process produces spiraling of the dermatomes, so the sensory regions, and it's also important to remember when you're looking at defects of the dorsal ventral axis. So limb patterning can be quite complex and hard to get your head around because there's lots of terms and signaling molecules. But on the slide, I put the key molecules and signaling centers, so hopefully that will be helpful to you. I'm going to give a quick run through of things, but feel free to come back and look at the slide. So in terms of specifying if a limb is upper or lower limb, Hox genes and TBX transcription factors are really important. So TBX4 is expressed caudally, and when it's expressed, you get a lower limb, whereas TBX5 is expressed rostrally, and when it's expressed, you get an upper limb. The positioning of limbs at specific levels in the body is important to ensure correct development. And Hox genes and FGF10 are the signals identified as playing a key role in positioning growth of the limb bud and therefore the limb. So a mutation in HOXB5 causes the upper limbs to grow more rostrally, so higher up than they should, and this leads to a condition known as Sprengel's anomaly, which can be pictured on the slide. So after the limb bud has formed, the limb is patterned in 3 axes, and each axis has its own signaling center. So the signaling center of the proximal distal axis, so from like the shoulder down to the tips of your fingers, is the apical ectodermal ridge, or AER. It's thought that increased FGF or fibroblast growth factor signaling from the AER induces more distal structures. And several birth defects are linked to issues with this signaling center. So these include amelia, in which there's no limb formed, and phocomelia, which is a specialized type of limb loss where the distal structures remain, but the proximal structures are gone. And this is pictured on the slide, and it was linked to the thalidomide crisis. So next up is the rostral caudal axis, and the signaling center of this is the zone of polarizing activity, also called the ZPA for short. And this is found on the caudal aspect of the developing limb bud. It releases sonic hedgehog, or SHH, and that acts as a morphogen to pattern. So there are several developmental defects linked to this axis, all linked to different number of finger development. And so these are polydactyly and syndactyly. Okay, and the final axis is the dorsal ventral axis, and the signaling center for this is actually the dorsal ectoderm. And it works by releasing Wnt7a that's not released by the ventral ectoderm. And this in turn induces something called LMX1 activity, so that's a transcription factor, in the dorsal mesenchyme. And mutations to that transcription factor can lead to a condition called nail patella syndrome. Nail patella syndrome basically means an individual lacks a patella and have highly reduced nails. As before the rotation of the limbs, all of these structures lay on the dorsal side of the axis. Okay, next up is heart development. Again, quite a complex process with several steps, but we're going to break it down. So it starts with induction of the cardiac mesoderm, and that's derived from the lateral plate splanchnic mesoderm, and the key signal here is BMP2. Next up, you get formation of something called the cardiac crescent. So this is made of the cardiac fated cells, and they migrate to form these heart fields, and the heart fields then fuse. Then the cardiac mesoderm starts to form something called angiogenic clusters, and then these clusters form the left and right endocardial tubes. And the key molecule here is VEGF. The next step depends on, or is reliant on, embryonic folding. So rostral folding brings the developing heart caudally, so it now lies underneath the developing brain. And lateral folding brings this left and right heart tube that have developed separately together, and they fuse in the midline to form one linear heart tube. This one linear heart tube consists of, from rostral to caudal, paired dorsal aorta, truncus arteriosus, bulbus cordis, primitive ventricle, primitive atrium, and paired sinus venous. And at this point, the myocardium has migrated to the tube, so the developing heart now has some contractile activity. The next step is something called looping, and this moves the presumptive chambers of the heart to the correct spatial regions, because up until now, the atria have been lying inferiorly to the ventricles, which is not how they sit in the developed human. So the primitive atria in looping move superiorly and dorsally, and the primitive ventricles move inferiorly and ventrally. At this point, the left-right asymmetry is also quite clear and apparent because the majority of structures of the heart are lying on the left-hand side of the body. The next stage is septation. So prior to septation, the heart is still this one linear tube, and septation therefore needs to occur so that the heart can be divided into the four chambers that we have. So endocardial cushions are the key structures in septation of the heart. They are formed from something called cardiac jelly, and they also have cells that have migrated into them. They are in two locations, so the outflow tract and the division between the atria and the ventricles. In the outflow tract, the cells that have migrated are those neural crest cells that we talked about, but in the atrioventricular ones, it's actually mesodermal cells. So we're going to first talk about separating the atria from the ventricles. And this is done as the endocardial cushions within that region fuse together. And this results in the formation of two atrioventricular canals on each side of the heart. And the septation is completed by the formation of the mitral and tricuspid valves that form in these canals. Next up is atrial septation, so separating one atrium into the left and right atrium. In this process, a septum grows downwards, so from the top of the heart. In embryonic development, this is an incomplete septum because it contains the foramen ovale, which acts as a valve in fetal circulation, enabling blood to flow from the right atrium to the left to bypass the pulmonary circulation. And then if this continues on into adulthood, then you get atrial septal defects. So next up is separating the ventricle, so into the left and right ventricle. The interventricular septum is actually made up from two different structures. So the first is a muscular part that grows up from the apex of the heart. The next part is the intermembranous part, and it forms later as part of outflow septation. And interventricular septal defects mainly occur within this membranous part of the septum. The final septation is outflow tract septation. And this occurs by fusion of the conochocal ridges formed from the endocardial cushions present in the outflow tract. The hemodynamic forces that are occurring in the outflow tract are really important in this septation, and it's the spiraling of blood that results in the spiraling septation of the two vessels as the truncus arteriosus divides from the inside into the aorta and pulmonary artery. The outflow septum fuses with the septum intermedium and muscular interventricular septum, and this basically ensures that the correct outflow tract is linked to the correct ventricle, so the left ventricle to the aorta. This process can go wrong given its complexity, and one key congenital defect that you need to know is tetralogy of Fallot, and this is caused by issues with outflow and therefore ventricular septation, and it leads to four key features, which are pulmonary stenosis, an overriding aorta, ventricular septal defects, and an enlarged right ventricle. There are several issues with outflow tract septation, including truncus arteriosus, in which there's no division at all, and transposition of the vessels, in which there's a mismatch between ventricle and outflow tract, so the left ventricle going into the pulmonary artery, for example. Now, circulation changes quite a lot between in utero and postnatally. And this is because on the first breath, the lungs expand, so the pulmonary circuit starts to be used. And after birth, the baby no longer receives its blood supply from the umbilical cord. So that's the reason underlying the changes that occur. So the changes themselves are umbilical vessel closure, ductal venous closure, so that in the uterus allowed the liver to be shunted past. And then as the pressure in the right atrium falls, below that in the left, the foramen ovale that we talked about closes as well. And then finally, the ductus arteriosus that connected the pulmonary artery to the aorta to bypass pulmonary circulation closes, and it closes due to a rise in bradykinins and prostaglandins. So the next system is the urinary system, and the kidneys develop from the intermediate mesoderm. The kidneys develop craniocaudally in 3 stages: pronephros, mesonephros, and metanephros, but metanephros are the true kidneys that we have. So development of these metanephros begins at the end of week 4, and they develop from 2 key structures. So the ureteric bud, which is an outpouching of the mesonephric duct, so that's linked to the previous stage of kidney development, and a bunch of mesenchyme turn the metanephric blastema. And these structures come together. The mesenchyme induces the bud to undergo branching morphogenesis. So it branches and it branches until the pelvis, calyces, and collecting ducts are formed. And then the tip of the collecting duct induces the mesenchyme surrounding it to form the nephrons. Now, the metanephric kidney develops a lot more caudally than the kidneys sit in the adult human. And to reach their position in the adult body, they therefore need to ascend. As they do so, there are changes in blood supply to progressively higher segmental arteries. But this process of ascension does not always go correctly. Sometimes it just doesn't happen, leading to a pelvic kidney. But another case is something called a horseshoe kidney, in which rather than 2 separate kidneys, one fused kidney develops. So as this fused kidney tries to ascend, it gets stuck on the inferior mesenteric artery, so it ends up a bit more caudal than it would normally be. So then you have your kidneys that are formed. Now you need the rest of the urinary system to form. So the bladder forms from the allantoid, which is an outpouching of the hindgut, and also the superior part of the urogenital sinus. The inferior part of this sinus forms the urethra. Now, in the initial stages of development, the cloaca that we talked about before acts as an opening for both the urogenital and gastrointestinal systems. Obviously, that's not how it occurs in the adult human, and so this needs to be separated. And so the separation is done by the urorectal septum, so you have different openings for the urogenital and gastrointestinal system. And then the bladder is connected to the kidneys by the mesonephric ducts, so the mesonephric duct form the ureters. The next system is the genital system. And we're going to break this down into several different stages. So we're going to start with chromosomal sex, then move on to gonadal, and then phenotypic sex. So chromosomal sex is typically thought to relate to X and Y. So as humans, we have 46 chromosomes in our diploid cells, 2 of which are sex ones, which can be X or Y. XX normally leads to a female sex, XY to male. So the Y chromosome normally leads to the male phenotype, but it's not always the case. And it's actually one key region on the Y chromosome that's really important, called the SRY gene. That stands for sex determining region of the Y chromosome. And it encodes a DNA binding protein, the exact function of which is not known, but it could work through either switching off DAX1 expression or switching on SOX9 expression. Next up is gonadal sex. So this is, do ovaries or testes develop? Now, the gonads develop as a ridge of intermediate mesoderm, and this ridge can fail to develop, and that will lead to an agonadal state, so someone without testes or ovaries. At the very start of development, the gonadal ridge looks the same in both males and females, so there's no obvious difference between the 2 sexes. Then the SRY gene expression is turned on if it is present, and it causes cells of the mesoderm to differentiate into Sertoli cells, which direct testicular formation. When the gene is not expressed, the cells differentiate into granulosa cells, and that dictates ovary development. Now, the somatic cells of the gonads are associated with the germ cells at this point, and the germ cells are those that develop into the sperm and the egg. So the next type of development we're going to talk about is phenotypic sex, which can be further divided into internal genitalia and external genitalia. So in terms of internal genitalia, in the indifferent stage, so when you can't tell the difference between the 2 sexes, both sexes have a paramesonephric, also called Müllerian, and mesonephric, also called Wolffian duct. And in the female, the Müllerian duct is retained to give the uterine tubes, uterus, and part of the vagina, and the Wolffian duct degenerates. In the males, it's the other way round. So the Müllerian duct degenerates, and the Wolffian duct differentiates to form the epididymis, vas deferens, and seminal vesicles. And in the males, there are two key hormones that are important to ensuring this process. So testosterone acts to maintain the Wolffian duct, and anti-Müllerian hormone causes the Müllerian duct to degenerate. So the lack of expression of those two hormones in females is why you get female internal genitalia. Then in external genitalia is also a hormone-dependent process. Now, testosterone can act as a hormone, but for many of its actions, it's actually converted into 5-alpha dihydrotestosterone by an enzyme called 5-alpha reductase, and this leads to an almost stronger form of testosterone. So for the development of penis, scrotum, and prostate, you need this strong form of testosterone to be present. So the next system I want to talk about is the gastrointestinal system. So as we mentioned, the endoderm is really important to this, and the formation of the endodermal gut tube was described when we talked all the way back in embryonic folding. So at this point, the gut tube is suspended by the body by a sheet of mesoderm, now we'll term the dorsal mesentery. At the level of the stomach and liver, there's also a ventral mesentery, which becomes the lesser omentum and the falciform ligament. Now, all organs in the GI system are formed by interactions from the surrounding mesoderm and the endodermal epithelial lining. And the system can be thought of as three parts linked to embryonic development. So you have the foregut, the midgut, and the hindgut. So the foregut is up to the mid-duodenum, the midgut is up to the swelling factor, and the hindgut is everything else. Of note, the midgut expands a lot during embryonic development, and as it does so, it rotates 270 degrees while fixed to its artery, which is the superior mesenteric artery, and the vitelline duct. Now, this rotation is split into two parts. So you have 90 degrees on herniation and 180 degrees when it returns to the abdomen, because its expansion is so great, it leaves and then comes back. And then in terms of specific organs of the gastrointestinal system, I first want to mention lung development. So whilst not strictly part of the GI system, they do bud off from the developing esophagus and then undergo branching. And if there's abnormal separation of this esophagus from the respiratory system, then you can get a fistula, and this can lead to a lot of complications postnatally. Next up, I want to talk about the liver. So the liver develops as a diverticulum from the gut tube. And this diverticulum, this budding or pouch, has a cranial and caudal part. The caudal part forms the gallbladder. And the liver bud and the vasculature that will supply the liver pass through the lesser omentum within the free margin and continue to do so into adulthood. The pancreas is the next organ of note, and this develops as two buds. So it has a ventral bud, which is part of the liver diverticulum, and a dorsal bud. And as the gut rotates, the ventral bud moves behind the duodenum to fuse with the dorsal bud to form the hook-shaped pancreas, so to form one organ. The ventral bud forms the uncinate process of the adult pancreas. But this process of rotation, as with other processes of rotation we've discussed, can go wrong. And it can lead to either accessory pancreatic ducts, or more importantly, an annular pancreas, in which the duodenum can be surrounded by pancreas, and that can lead to obstruction of flow through it. And the pancreas, during this process, becomes secondarily retroperitoneal. There are other parts of the gastrointestinal system that become secondarily retroperitoneal in development, and the key ones to remember are the ascending and descending parts of the colon. Okay, the final bit of development that I'm going to talk about today is head and neck development, which is linked to something called the branchial arches, that are also known as the pharyngeal arches. And in terms of evolution, these are linked to gill development in fish. So as humans, we have 5 branchial arches arranged in pairs. These are 1, 2, 3, 4, and 6. There is no 5th arch in humans. And the general structures of all the arches is the same, and it's as follows. So you have the pharyngeal cleft made of ectoderm, which is the outermost structure, seen in yellow on the picture. You have a mesenchymal core in the middle that's made of mesoderm and those neural crest cells that we talked about earlier. And the pharyngeal pouch made of endoderm is the innermost structure, and that's seen in blue. Each arch gives rise to a number of structures, which includes a rod of cartilage, muscle, nerve, and artery, but the exact structures do depend on the arch. Now, it's important to note that this cartilage that can become bone, if you remember, is neural crest cell derived because the bones of the face are from neural crest cells, which is unlike anywhere else in the body where they are mesodermal in nature. So the key structures are on the table for you to read through in your own time, but I just wanted to draw your attention to one or two. So firstly, as you can see, arch 1 is important in palate development. So if you have issues with arch 1 development, you can get cleft palate and facial clefting. And then also in terms of the arteries, initially each arch is supplied by a separate artery, but these remodel, leading to either regression of them or formation of arteries that are seen in the adult. So the arteries supplying 1 and 2 regress, the arteries supplying 3 becomes the carotid arteries, and those supplying 4 and 6 become the aortic arch and also the pulmonary trunk. Okay, that's all of the information to cover. I appreciate it was quite quick, but I have a few questions coming up, and you could just put the answer in the, either say it out loud or you could put it in the chat if you feel more comfortable doing that. Okay, just to check if you have taken anything away. So question 1 is, what germ layer is the stomach epithelium derived from? If you can't remember, that's also okay. So the answer for this one is A, endoderm. So the endoderm is the germ cell layer that gives rise to the gut tube lining. Yeah, let's just remember that one. Next up. Oh, okay. What is the name of the vessel that will close at birth to prevent blood flow between the aorta and the pulmonary circulation? Almost. So the answer is C, that's right. So the foramen ovale A, which was close, separates the left and right, or allows flow between the left and the right atrium, but it's also to bypass the pulmonary circulation, whereas it's the ductus arteriosus that connects the pulmonary artery and the aorta to bypass the pulmonary circulation. Okay, and the last one. Ooh. Which of the following muscles are derived from the second pharyngeal arch? Now, I appreciate we covered this quite quickly, so you might not know, but I'll just bring it to your attention anyway, because this has come up in exams before. Yeah, you're right. It is the muscles of facial expression. So the muscles of mastication are actually, if we go back, linked to arch 1, and the pharyngeal muscles are arch 4. Okay, well, that's everything from me. We'd really appreciate it if you could provide some feedback. So you can either scan the QR code, or I can try and put the link in the group chat, in the chat, whichever is easiest. And if you have any questions, feel free to either email or you can ask now. I'll stay on for like a couple of minutes.